Welcome to Conversations. I'm Don Leopold, Distinguished Teaching Professor at SUNY ESF. The American chestnut once dominated the eastern forest of the United States, not only in size, but in numbers. In the past century, this tree was decimated by an invasive fungus. By the mid-1900s, the American chestnut trees all but vanished. For the past 2 decades, SUNY ESF professors Dr. Charles Maynard and Dr. William Powell have focused their research program on an effort to produce a blight-tolerant American chestnut tree. Applying cutting-edge biotechnology, their efforts have yielded such a tree. Their discovery holds great promise for the restoration of this once prominent tree species to the eastern forest. I'm pleased to welcome one of the collaborators in this project to Conversations, Professor Bill Powell. Welcome, Bill.
Thank you, Don, and I appreciate this opportunity to talk about our chestnut project. I'm very excited about it.
Well, the American chestnut is just one of hundreds of tree species native to eastern U.S. forests. So why is there so much interest in this single tree species?
Well, in your intro, you can see that it was a very important tree to the ecosystem of the eastern forest, but it also has a special place in the hearts of the population in this area. It's kind of immortalized in song this time of year. You hear chestnuts roasting on an open fire, or this sleigh ride song where it goes chestnuts going pop, pop, pop. So it has a unique place in our culture, and has a neat story to it too. So it was once a very important tree, but we have lost that tree, and it's all because humans were starting to bring over Asian chestnut trees in this country. And that was about a century ago, but at that time they didn't realize that when you brought a tree from another part of the world, you don't only bring that tree, but you bring all the microbes on that tree. And it turns out there was a fungus on that tree that would devastate our American chestnuts here in the United States. And so we got that story of the loss of the American chestnut, and now we have the opportunity, using the newest techniques around, to bring back that iconic tree.
So in terms of its importance, a lot of trees are just thought of as simply economically important for lumber. What else is it about the American chestnut?
Well, it's definitely important for lumber because it has very rot-resistant wood, but it also produces a very significant nut crop or mast crop for wildlife, which is very consistent from year to year, unlike the oaks that have since kind of replaced the American chestnut. The chestnuts also were very important to the rural farmers. There's our William Bray wrote one time that local farmers would realize more income from chestnuts than from any other farm product. So you can see it was very important. And the neat thing is these were wild trees. They weren't orchards of trees. So the farmers would go out and just collect them, and it's like manna from heaven, it's free food that they could sell. It was also important for the Native Americans here. They were actually utilized American chestnut quite a bit, and it was kind of interesting. The local Native Americans, kind of when we lost the chestnut tree, was the same time they were losing their language, and it kind of parallels that loss, and we hopefully that will parallel the regaining of those things too.
And just how abundant was this tree in the eastern forest?
So within its range, and I have to emphasize within its range, which is along the Appalachian Mountains, it was probably on average 25% of the standing timber. Okay, so 1 out of every 4 trees would have been an American chestnut. Of course, some places would be stands of 100% American chestnut, and others less. And then it extended out from there. So when we say it's one of the most abundant trees in the eastern forest, really we're talking about in its native range. Outside, it was still a predominant tree, but not necessarily the most abundant one.
But how abundant was it in New York?
In New York, I would say, at least in the southern tier of New York, below the Adirondacks, it would probably have been about 1 out of every 4 trees, yeah.
So scientists have tried for decades to make American chestnut disease-resistant through hybridization. How well is this approach working, and what are its shortcomings?
Okay, so there has been many attempts over the past century. Originally they tried to do fire breaks to try to stop the blight. That did not stop it because it can also live on oak trees, and therefore it can just bounce right over those fire breaks. They tried to use fungicides, and they did not work. They tried breeding programs. If you go out and try to buy a chestnut tree today, most of them are going to be hybrid chestnuts crossed with Asian species, which are naturally resistant to blight. And those trees actually are not bad trees if you want to use them for agriculture or ornamental uses. But for a restoration tree, they do not work because the Asian chestnut trees have been bred for for centuries to be an orchard tree. and their maximum heights might be 40 to 50 feet, where an American chestnut gets up to 80, 90, 100 feet, maybe even 110 feet in height. So American chestnut was really a canopy tree. So when you make hybrids with the Asian species, you tend to get a shorter tree, and therefore it will not really survive or replace what the American chestnut was in the past. So
you mentioned the fungus gets on oaks. Is there any concern about oaks becoming susceptible to the disease?
Right now, it's either lives on the oaks as a saprophyte or as a very minor pathogen, such as scarlet oak. If scarlet oak is in a drought situation, it could get some damage from the chestnut blight fungus, but generally it just lives as saprophyte.
So what does that mean, a saprophyte?
So it means it's just living off dead tissue, basically. So living off the dead bark on the tree, just sitting there. And the important thing about that is that even if we were to get rid of all the American chestnut trees and then try to bring them back later, the fungus is still going to be there. You can't get rid of the fungus. It's still going to be on the oaks and come back to the chestnut.
Well, so I talk a lot about trees around the country every year, and when people find out about your project, they want to know, how did you come up with this solution to this problem?
Well, it's really kind of an interesting story. When I was in graduate school, I worked on the American chestnut, but I worked on the fungus side. So we were working on what's called hypovirulence. That means reduced virulence of the fungus, and it was caused by a virus that infected that fungus. Well, while I was studying that, I learned that one of the major virulence factors, the way the fungus attacks a tree, is to produce an acid called oxalic acid, or the salt oxalate. And I always kept that in mind when I came here and started working on the transgenic approach to American chestnut tree restoration. And it turns out that one time one of our postdocs brought back a book of abstracts from a meeting, a plant physiology meeting. And she gave it to me so I can kind of see what was going on. And I was reading through it, like I usually do, and I came across this one article, or one abstract, that said these people were putting this enzyme in tomato to make it resistant to a fungus. And that particular enzyme was oxolate oxidase. And I made that connection right there. That was kind of the eureka moment. And that's what scientists kind of do. They kind of make connections. And we found, and what it is that this enzyme will actually break down that acid, oxalic acid, and detoxify it. And if you could do that, then the fungus no longer is able to attack the tree. So we immediately got a hold of this particular gene for this enzyme and started putting it into the American chestnut.
So to what extent is your discovery hard work versus luck?
It's a little bit of both. There's always some serendipity in research, but it's really, though, a lot of hard work. The whole idea of putting a gene into chestnut, we had to develop the techniques to regenerate a tree from a single cell. And that was mainly done in Dr. Maynard's lab, and took about 16 years, really, to get that whole system up and running efficiently. Then we had to test genes out, and we did not only test the oxalate oxidase gene, but we tested several others, and to find which one works best, which was the oxalate oxidase.
Were there points along the way that you thought this was not a solvable problem?
I never thought that. The only thing I've ever thought is, how long is it going to take to solve the problem? It's just a matter of getting the right combination of genes and the techniques together to produce a tree. So I always thought it was going to be possible. It just took actually a lot longer than we thought. When the American Chestnut Foundation originally approached us, they asked us, well, can we take this new approach, this genetic engineering approach, as opposed to breeding? And we said, yes, we think it would take 10 years. And then it took us 26 years.
Well, I think they're probably happy that they waited. So can you provide an overview to the process in relatively simple terms, if that's possible?
Right. So we found a gene that we thought would work, the oxalate oxidase. To get the gene in, we use what's called Agrobacterium tumefaciens. It's a bacteria that's a natural genetic engineer. It puts genes into plants in nature. And what scientists have done is basically tame that bacteria, so that instead of putting genes it wants to put in, it'll put the genes we want to put in. So we put the oxalate oxidase gene into the Agrobacterium. We use that to transform cells of the American chestnut, and we have to use what's called somatic embryos, which are little embryos that bud off new embryos. That was a long process developing that. And we worked with other collaborators, such as Scott Merkel at University of Georgia. And so we get those in there into single cells. We regenerate a whole plant, and all along the way, we have to keep testing the plants. We usually have to make hundreds of these to get a few at the end of it. We regenerate the embryos into shoots, get the roots on the shoots, get the plants acclimatized so they can go out into the environment, and then we test them for blight resistance.
Would there be an easier tree species to work with given the process that is involved?
Absolutely. Matter of fact, hybrid poplar is a much easier tree to work with. You can actually regenerate shoots from just leaf slices. And in fact, we used poplar during this whole process. That was the first place we tested the oxalate oxidase gene. We transformed hybrid poplar and found out we could actually enhance resistance to Septoria muciva, which is a leaf and canker disease on poplar. And so we kind of did that first because we were still developing the transformation methods for the chestnut. So it gave us a proof of concept that this would work also in chestnut.
Can you tell us a little bit about this gene that you're using?
Okay, the gene oxalate oxidase is actually a very common gene. It's found in almost all grains and a lot of dicots also. And it's a gene that basically used for defense. There's a lot of fungi out there that produce this oxalic acid as a way to attack the tree. And the trees have evolved these ways to defend it. This gene, oxalate oxidase, there's other ones. There's an oxalate decarboxylase, and there's other multiple biochemical pathways to get rid of the acid. But this one is one of the simplest ones. So, what we're moving into the tree isn't something that's kind of strange and unusual. It's something you eat all the time. In fact, if you ate a bagel this morning or a muffin, you ate oxalate oxidase.
Okay, so this is completely natural.
Yes.
Not anything scary.
Nothing scary. And in fact, that's one of the reasons why we wanted to choose this particular gene, because it isn't something that's unusual, that people aren't familiar with, even though they might not know they're familiar with it, they actually are.
So how resistant to disease are your selections?
Okay, so the way we test for resistance, we have a standard Chinese chestnut control that we use. We use that one called King, and or Qing. And what we do is we look at either in leaf assays or in stem assays how big the canker gets compared to that. And our trees basically are as resistant as our Chinese control, and in some instances it looks like they might even be a little bit more resistant than the Chinese control. Now, Chinese chestnuts vary in the amount of resistance, so we just kind of like say we're at least equal with Chinese and possibly more resistant depending on which one you compare it to.
So this time of year you see a lot of chestnuts at markets and in the grocery store. What chestnut is that?
They could be different ones. If they come from Michigan, they could be hybrid chestnuts because they have a small industry starting in Michigan. But actually, most of them are, in the East probably come from Europe, so they're probably European chestnut. They could be Chinese chestnut also. And those are those very large chestnuts that you see in there. American chestnuts are actually smaller than those chestnuts. I always compare them, the Chinese and European chestnuts are about quarter to $half size, where the American chestnuts are kind of like dime to nickel size. So they're a smaller chestnut. It's claimed, and I agree with it, they are have better flavor than the others. But yeah, so you can't actually buy an American chestnut in the store right now.
Okay. So what are the most common concerns that have been expressed about bringing American chestnut back to the eastern forest by using your approach?
Okay. Well, obviously there are people who are kind of skittish of using genetic engineering, and that is fine. And when it first came out, 30 years ago, I can understand that, but we've had 30 years of safety using this type of technique, so I don't think that's a problem. In fact, by putting in a single gene into the American chestnut tree for resistance, it's actually much safer than doing hybridization, where you're bringing in literally tens of thousands of genes, and you don't know what's going to happen with those. In fact, sometimes we get male sterility from hybrids. Sometimes you get things like internal kernel breakdown from hybrids. You don't see that when you're just putting in one single gene at a time. So that's one concern, is people don't like the technique too much, but some do. Some are very enthusiastic about it. But there are also people who kind of worry about bringing the tree back that's been gone for 100 years. I don't think that's a problem, because 100 years in the scheme of evolution and stuff is actually a very short time. So bringing it back after 100 years, I don't think that's going to be anything but beneficial.
Sure. So was there American chestnut in New York 10,000 years ago, for example?
I'm not sure 10,000 years ago. I have to check when the last ice age was.
Well, it's when the ice retreated. So so there's a good example where American chestnut probably was not in New York State. It's
absolutely. So actually, when the glaciers receded and stuff, American chestnut was one of the last of the hardwood species to repopulate as it moved northward. So it doesn't spread very fast. So that's another thing that people shouldn't be too scared that this is going to be like a weed and it's going to go everywhere. It actually moves very, very slowly. Usually only 1 to 2 kilometers. They've done studies 1 to 2 kilometers every century. So it's it's really going to rely on people planting these to get them out into the forest.
So are there any examples of other trees in particular where this genetic modification process has been used to give us resistant to disease, especially types of trees?
Okay. So with the American chestnut, we will be the first for a restoration tree. No one has done that with a restoration tree. Some people have done that with orchard trees. There is a virus-resistant plum that has been approved and is out there if people want to plant it. And there's some things like the non-browning apple out there. There's a big effort right now to engineer orange down in Florida because of the citrus greening problem they have down there. But there's not too many. So we're kind of blazing the trail, especially in the area of restoration.
This technique you're using, are there other tree species that might benefit from it if you tweak the process a bit for those species?
Absolutely. So when the blight went through, it not only killed American chestnut trees, but it actually killed off a lot of related species, such as the Chinquapin. And the one that was most devastated was the Ozark Chinquapin. And since it's the same fungus, and it's a closely related tree species, we could actually do the same thing for Chinquapin that we did for American chestnut and bring that tree back from near extinction.
Are there other tree species unrelated to chestnut in the eastern U.S. where this approach generally might be useful?
Yeah, so if you're just thinking about the general approach of genetic engineering, of moving in genes that will confer resistance, there's a lot of trees. I mean, obviously the one would be the American elm. We actually are starting to work with that. We worked with it some in the past and put it on the shelf because we focused on chestnut, but now we're getting back to it. So American elm would be one. There are a lot of diseases out there. There's thousand canker disease of walnut that's starting to spread. And of course, everybody's familiar with emerald ash borer, hemlock woolly adelgid. So there's a lot of trees that could benefit from these types of techniques.
So what are your next steps?
So our next steps are, we're actually going right now through what's called the regulatory process. We can't distribute our trees until we get approval from the federal government. And so we're working with the USDA, the EPA, and the FDA, all three agencies. It's kind of exciting. Just a couple weeks ago, we submitted our application to the USDA. And so that process has started. And right now, we're just waiting for the first review, which is just a technical review to make sure we have everything in there properly. And then we go through the actual science review. And during that time, they're going to have an open comment period from the public, which I hope everybody who watches the show will write in. If you want a chestnut tree, this is the time to actually do something. Write in and say you want a chestnut tree to the USDA.
So we're all trained as scientists, and we're trained to focus in the lab. And is there something about this whole program, this research program, that has you've had to reach back and develop other skill sets that maybe you didn't think about as a graduate student?
Well, absolutely. I mean, no one trains you in the regulatory process, which is not a science-based process anyway. It's more of a political type of process, I would think. Really, the past 2.5 years was learning what is needed and making sure we have all the experiments done. We're doing like insect feeding the leaves. We've fed pollen to bumblebees. We've looked at, even had frog tadpoles eating the leaves and stuff, and looking at mycorrhizae on the roots. Getting all of that together and put it in the right format for the regulators to review. So we had to kind of learn what they needed and then start doing it.
So when this tree is finally approved, most folks probably won't understand all the background that led up to that approval, but it's a fairly rigorous process.
It is a very rigorous process, and it's long. I mean, we're excited about just submitting to the USDA, but it's really going to take them about 2 years, close to 2 years to get that approved. And so we can't actually pass out trees until that process is finished, as well as later we need to get the the FDA approval. We're still talking with the EPA if they actually regulate us, because this is not a pesticide, so we're trying to figure that out. And then finally, we'll probably go up to Canada and go through their regulatory system, because chestnuts do actually go up into Canada.
So when the tree's finally approved for release, how can people acquire a disease-resistant American chestnut?
So we're going to be working closely with the American Chestnut Foundation on distribution, but basically we kind of see our work here at ESF as pollen distributors, okay? Because one of the things we want to do is build in as much genetic diversity as we can. So what we'd like to do is have our trees outcross to as many wild-type trees and rescue their genotypes and get it back into the population. So we're going to be big pollen distributors. So that one way you could do is you can plant a mother tree, or find a mother tree on your property, and then we could distribute pollen to you, and then you can actually get a lot of offspring from that, such as this one right here. That's one way. We also will be distributing some seedlings to people, and as well as some scions for grafting. And we're going to do this all as a not-for-profit. I should mention that we have not patented these trees, so we didn't want a patent to slow down any type of restoration, and so we're going to do this as cheaply as possible for the restoration.
So can you show us the seed again, Bill?
Yeah.
So are you able to establish mother trees now? It's not too early to do that, at least.
No, actually, we have a program with the New York chapter, and actually the National is starting to do that. Also put in what they call conservation orchards, or individual trees in people's yards and stuff. So basically, the female flowers follow the male flowers by about a couple years. So we really need to get those trees out there and get them mature enough, which will be about 4 or 5 years to get those flowers, and then we can outcross them. Right. And this nut here is just an example. This is actually a second generation outcross. So we took our original trees that we made, we outcrossed them to wild type American chestnut, took those seed, we grew them up, made more pollen, and this is actually the result of the second generation. And this is the kind of pollen that will outcross, so then what people will be getting is a third generation outcross. Okay.
So these trees won't probably be resistant to disease or fully resistant, but they'll provide pollen before they succumb to the next blight.
The mother trees will be just typical wild-type American chestnut trees, so they will die. And so the quick trick is to try to keep them alive long enough that we can cross with them and get their offspring. When you get the offspring, half the nuts will actually inherit the gene, simple Mendelian genetics, but that half will be fully resistant. And we have a simple test that you can just do a little leaf punch and test it to find out which ones actually contain the gene.
Great. So how are some ways that viewers can help this whole process be successful? Okay,
so there are several ways. One I've already mentioned is once we go through the regulatory process, they always have an open comment period. We really want everybody who wants a chestnut tree to write in to the USDA and later to the FDA and possibly to the EPA to say that, well, we do want these trees. If they stop us there, they're not going anywhere. So we really need the public behind us on that. Another thing is the stages we're at right now is more of kind of a application of restoration, or it's not basic research anymore. So it's getting harder and harder to get funding from the federal granting agencies. So if you want to donate to the project, we always appreciate that. And you can go to our website at www.esf.edu/chestnut. And there's a place you can click on there if you want to donate directly to the project.
Okay. So we've talked a little bit about this grander ideas of this program is sufficient, I think, to really make its mark in terms of the best research in forestry in centuries maybe. But we've talked about this tree restoration center. For if there's someone out there who wants to have the most incredible legacy when it comes to saving trees, you want to talk a little bit about this tree restoration center?
Yeah, so I don't want this to be a one-off success story. I really like to see this as a foundation for a tree restoration center. And I think, our school is excellent for that type of opportunity. We have all these ecologists and people who can work in that area. So my idea here is that we'll use chestnut as a foundation, We'll go and pick some of the lower hanging fruit, like the Ozark Chinquapin and maybe the American elm, and then kind of branch out and be a place where people can ask us to, well, we need this tree rescued, can we do it, and how can we do it, and grow from there.
So I know you've, and you always acknowledge your very big army of people who have helped, including
Absolutely.
a lot of graduate students and some really talented undergrads, even high school students. Is this a place where graduate students might have an opportunity to continue to contribute to the program?
Absolutely. I've had, right now I have about 8 graduate students, and, I'm having 4 of them graduate, hopefully this year. So there's always going to be room for more. And like you said, we have anywhere from 4 to 12 undergraduates work on a project every year, and high school interns during the summer. We had a lot of those come through. So if you want to work on a chestnut project, this is the place to do it. Yeah.
It just seems that something this spectacular in importance happening at ESF seems just sort of bigger than ESF. What if you were someplace else? Why did this happen at ESF? Is there anything unique about ESF where this might have been the ideal place for this program to develop?
Well, I think it's because of our goal. I mean, the old thing of improve your world type thing. And really at ESF, I've been given the opportunity to go any direction I want. Okay, and early on in the project, there was a lot of naysayers. People saying that, well, this probably won't work. No one's ever made a plant fungal resistant before. No one's ever engineered a forest tree. All these type of things that, oh, this can't be done. But here at ESF, I was able to pursue that, even when the odds are low, and it turned out to be a good success, a big success. And I don't think it could have been done anywhere else, really.
So could you tell us a little bit about Dr. Maynard's role in all this the last 30 or so years?
Yeah, Dr. Maynard, we've been co-directors for the first 25 years of this project. And originally, and we worked very closely with the New York chapter of American Chestnut Foundation. Actually, 3 of their members came to us back at the very beginning, back in 1989, and asked us if we can do a different project where instead of using breeding, we use genetic engineering. And came to Dr. Maynard and I, Chuck, and it turned out that our skills really complemented each other. Chuck had a background in forestry and in tissue culture. My background was in molecular biology and plant pathology. And so we kind of meshed really together. Chuck was really implemental in getting the tissue culture and the transformation system up and running, which we could not, we could find all the genes we want if we couldn't get the gene into the plant. So that was key to the success of this project.
So professors eventually retire.
Yes.
And what happens when that happens in terms of this project, this program?
Yeah. Well, initially it throws a lot of more work on me, but I'm really hoping that we will start getting more and more faculty in this area, and we can branch out to other species. And I'm hoping, in this tree restoration center we'll be part of that.
Great, great. Anything else you want to tell? You've been asked so many questions about American chestnut over the years. Is there something that you wish somebody had asked you, something that would be helpful for the public to know about?
Just that, I think we pretty much covered everything because it's such an important tree, but I think people need to realize is they are the ones that are going to actually do the restoration. Okay, we're a university. We can only produce so many trees. But it's really the public who's going to get these things into the forest. I mean, there was, over 3 billion of these trees in the forest at one time. How do you get to those numbers? You don't do it from a university or even from the American Chestnut Foundation. It's actually individual people who are going to have to go out there and plant these trees and propagate these trees. So I really want to get everybody on board if they want to get this tree back.
Could you tell us again about that website where viewers can go to find out more?
So we have a website here at ESF. The website is www.esf.edu/chestnut. So very simple.
Well, Bill, I want to thank you for joining us today. Very interesting discussion. And I want to thank the viewers for joining us as well. And hope you'll especially pay attention to some of the opportunities that you have to make this program more successful from this point on. So thank you for joining us today.